Hello students, in this video I am going to discuss about assisted reproduction technologies or ART. What is it? Assisted reproduction technologies or ART include a number of special techniques which assist infertile couples to solve their infertility problems. These technologies include IVF, gifts, etc. etc. So now what is it again? It is nothing but assisted reproduction technologies or ART include a number of special techniques which assist infertile couples to solve their infertility problems. These technologies include first is IVF ET, which means in vitro fertilization and embryo transfer. In vitro fertilization. Embryo, ET means embryo transfer. In short, test tube baby. So first is IVF ET. What is it? This is the technique of fertilization of an ovum with sperm carried out outside the body of female in a glass container. That means in vitro fertilization. So this is the technique of fertilization of an ovum with sperm carried out outside the body of female carried out outside the body of female in a glass container. That means in vitro fertilization. That means in vitro fertilization. Okay. Now come to the procedure. The procedure involves the following steps. The procedure involves the following steps. Number 1. Drugs are administered. Drugs, that means medicines, are administered to prospective mother, to induce prospective mother to induce or development of several ova, or development of several ova. That is superovulation. Now next, just before actual ovulation, the mature oocytes are retrieved. Okay. Now next step is the retrieved mature oocytes are placed in culture medium. Mature oocytes are placed in culture medium containing nutrients. The culture of oocytes is kept in incubator for about 4 to 6 hours. Semen to be used for insemination. Semen to be used for insemination is purified and concentrated by concentrated by wash and swim up technique. By wash and swim up technique. About 50,000 to 1 lakh fully functional sperm are taken. About 50,000 to 1 lakh fully functional sperm are taken. Sperm are taken. Okay. Now next procedure is purified semen is poured over purified semen is poured over the mature retrieved oocytes. Eggs are examined under the microscope for about 12 to 24 hours. Eggs are examined under the microscope for about 12 to 24 hours. 12 to 24 hours after insemination. Since fertilization occurs outside the female body, in a culture medium. It is called in vitro fertilization. The fertilized eggs show two pronuclei and a second polar body. The fertilized eggs show two macronuclei and a second polar body. Now next procedure is the fertilized eggs are selected and incubated for about 48 to 72 hours in an incubator. During this period of time, fertilized egg or eggs undergo cleavage and reach 48 cell stage. So now the fertilized egg or eggs are selected and incubated for about 48 to 72 hours. In any incubator. During this period, fertilized egg or eggs undergo cleavage and reach 48 cell stage. Now next procedure is the fertilized egg is stimulated to develop in vitro up to 32 cell stage. So now the fertilized egg is stimulated to develop in vitro up to 32 celled stage. Now this is the final procedure of in vitro fertilization. In this procedure, or in this step, actually this is the final step. In this step, this embryo is implanted on the endometrium of the uterus at 32 cell stage. The woman has now conceived and is pregnant. Further development of the child continues in the womb till it is born. Such a baby is called test tube baby. So now this embryo is implanted on the endometrium of the uterus at 32 celled stage. At 32 celled stage, the woman has now conceived and is pregnant. and is pregnant. Further development of the child continues. Further development of the child continues in the womb. Till it is born. Till it is born. Till it is born. Such a baby is called test tube baby. Test tube baby. So now here it is the process of test tube baby. In my next video I will discuss the another assisted reproductive technologies, that is GIFT, that means gamete intrafallopian transfer, artificial insemination and intrauterine insemination, etc. So now bye students.